Okay, thank you, Adam and Jacinta, and thanks everybody for coming along on a Wednesday morning tea time. Adam asked me to give a chat about my life's involvement with pelagic fish, and I was happy to accept. And so the talk is it's going to cover citizen science because it's Citizen Science Month. And of course, most of my work is involving with citizen science because I've dealt my whole career really with recreational fishers around the globe, and they've helped enormously with a lot of the research. Oops. For some reason, my slide is not progressing. Hang on. Okay. So just to start with, we're not the first people to catch marlin in Australia. I've got this bark painting hanging in my office, given to me by John Glaister, who was a Director of Fisheries in New South Wales. He was from the Northern Territory too. It's an actual painting of a black marlin, a bark painting from Elcho Island in Arnhem Land. And juvenile black marlin come close in there, and they're caught quite commonly. You can see them in shallow water, and there's no doubt that they were speared over a long period of time, and they're a totem in some areas. So we're not the first to be enthralled by the billfish. This is a potted biography of my fishy life. This will take about 5 seconds. I caught a fish when I was very little, and this was the first fish I caught. And I was always fascinated by fish and frogs and nature. A number of years later, I got some degrees from university and started looking for a job. Got a job too. It was with New South Wales Fisheries. It was a dream job. It was called marine angling biologist. I got paid to go fishing and work on species of fish that were important to recreational anglers. And a lot of the stuff we found out was unknown at the time, the life cycles of many of these fish, including the dusky flathead. So here's me a long time later with another flathead, but we did the whole life cycle of dusky flathead in estuaries in New South Wales, movements, reproduction, and that set me off on my career. But what really changed my whole life was seeing my first marlin jumping on the end of a line at Montague Island off Bermagui down in southern New South Wales. I just couldn't believe it. I went out with a commercial fisherman in a tinny, and he hooked up a black marlin, and it was just, I saw it jump, and I can still see it in my mind. Changed my whole life. In fact, I became obsessed with billfish and marlin. I think everybody realizes that knows me. And I've spent my whole career working in that general field and loving every minute of it. I was given responsibility early on in my career with New South Wales Fisheries. They just started off a tagging program, a game fish tagging program modelled on one that was going in the United States. It's operated by New South Wales Fisheries, still is. It started in 1973. That was a couple of years before I joined the department. So it's been going for more than 50 years. It's now funded from the New South Wales saltwater angling licence fees, and tags are sent all around Australia to every state and other neighbouring countries, all paid for by saltwater anglers in New South Wales, which is very nice of them. And this, of course, is this huge program, and in other parts of the world, tagging a fish is true citizen science. And we never used the term then, but it certainly is what it was and what it is. And the amount of information that's come from that is quite extraordinary. These are the species of fish that are eligible for tagging under that program. All of the billfish that we've just been talking about, sailfish, blue marlin, black marlin, striped marlin, swordfish, and spearfish in our part of the world. The tunas, the main species of tuna tagged are southern bluefin tuna and yellowfin tuna by recreational anglers. These are also tagged in quite large numbers, or have been in the past, by of commercial fishing vessels, because they're very commercially important. But the recreational tagging program has delivered a lot of information that was novel. Pelagic sharks are also on the list. Mainly mako sharks, shortfin makos would be tagged in the largest numbers, but also tiger sharks and whaler sharks in general. and what we call other game fish. So these are all pelagic fish or inshore fish that grow to a fairly large size. Each one of these fish in this slide, by the way, was a fish that was tagged and then recaptured. So most of these photos are when that fish was recaptured and then re-released. And we find that a lot of the recaptured fish are re-released. This is the history of the program. I said it's been going for over 50 years now. The little arrow there is when I was given responsibility for looking after the program. So it had just started. So we wanted this to really get some real science. So we really pushed in the game fishery at that time. Tagging was not that fashionable at all. And so we had a game fishing club that we formed at New South Wales Fisheries, and scientists and law enforcement people would enter tournaments and tag fish and encourage people to tag fish. And eventually it took off like a rocket, you can see there. And to the extent now that 95% of billfish are tagged and released that are caught in game fishing in Australian waters. And an average of around about 10,000 fish are tagged every year with tags. And you can see the recaptures go up and down too. The fluctuations often are due to the species mix that is being tagged, but also we found quite a big effect of El Nino, La Nina years as well. And this is the year that I left the fisheries department and formed my own research company, but I've maintained very close contact with the tagging program ever since. In fact, I've written the annual report of the program for the last 25 years. These are the numbers of the total numbers tagged, up for over 520,000 now, and approaching 10,000 reported recaptures. Billfish and tunas make up the bulk of these tags, but you can see yellowtail kingfish and sharks and tailing off-tube mackerels, and but significant numbers of all of these and recaptures of all of those. So it's not just billfish, but everything else. This is the amazing statistic. We're talking about citizen science. There's been well over 60,000 recreational anglers that have each tagged at least one fish. They've tagged those fish from probably approaching 12,000 boats now, individual boats, and representing over 950 fishing clubs. It's quite an extraordinary statistic. I'm just going to talk about just a little bit of the results, not much, but black marlin have been tagged in the greatest numbers. Black marlin Istiompax indica. I was talking to Adam the other day, and he said we'd like to know the Latin meaning of the meaning of the Latin names of these fish. Indica means from India, so the first black marlin was described that was caught in Indian waters. Istiompax is a bit tricky. Istio means sail, so that's the dorsal fin. The whole family is Istiophoridae, so that means sail bearer, and the sailfish, of course, that's the huge one. And Ompax, we're not sure, but it's got something to do with light, so it may be to do with the reflection of the light off the fin, but that's what I like to think it does. So in terms of mapping the movements of black marlin over many years, this is the recaptures we had, long distance recaptures these are, from the 1970s. So in the whole 10-year period, well, it started 73, so up until 1980, these were the long-term recaptures we had. Not many, but it was encouraging. As more fish started to be tagged and tagging became more popular in the 80s, we got more recaptures from out into the Pacific. And you see a lot of these are fairly remote places, and a lot were caught by, as I'll talk about a bit later, by artisanal fishermen and some fishing out of canoes and sent the tag back. This is the 1990s, where we got a lot more recaptures with a lot more fish being tagged over into Indonesia and the Philippines, and one that's crossed right over to Costa Rica. That's the record distance so far, tagged off Cairns. And in the 2000s, even more, up into a couple crossing the equator, up into the Northern Hemisphere, and one over to India. And then the last lot, you'll see that there's more tagging off Western Australia, where Jenny, mainly off Exmouth, Jenny Gates has tagged quite a lot, and you'll see that there's been some recaptures up through India and Sri Lanka. Just some record distances over the years that we've logged up. Black marlin, the one that went to Costa Rica, is the record, but you can see running down that list, I won't dwell on it. big distances of most of those species covering a lot of ground between oceans. I mean, the blue marlin, for example, was tagged off Sydney and recaptured in the Indian Ocean, so it must have gone up through off around Indonesian waters and made its way through to those waters and was recaptured by a Japanese longliner in the Indian Ocean. And you'll see right down the bottom, sailfish, that's by far the longest distance for the tagged sailfish. Most sailfish that we've had recaptured have been recaptured pretty well where they were tagged. In fact, we had a sailfish tagged off Broome, where they have a big sailfish fishery, and he was recaptured 7 years to the day later, 3 miles from where it was tagged. So not all of these fish are highly migratory, but and that sailfish probably went somewhere else in between, and we got some evidence that they do make an annual return to where we found them first. And these are the record times at Liberty on the program, quite significant for quite a few species running all the way down there. And we get very good information on growth rates from the tagging program too, not just where they moved, but how fast they grow, not just these long distance, long-term ones, but after 6 months, 12 months, etc. But it's not only recapture information in the tagging program that's important. The release information is very important too. This is a tag card that's typically filled in. So we know the species, the length and the weight. Sometimes these are estimated, sometimes they're measured. Mostly the weight is estimated. And and also we know the date that the fish was tagged, obviously, and the location it was tagged, either in the early days, just a name, like there it says Browns Mountain, but also the coordinates. And this is phenomenal. This is 520,000 individual fish locations and species and dates in the database. And so that data has been incredibly useful for looking at these sorts of things: catch composition through time, the size distribution of the catch by location through time. By looking at seasonal progression of sizes through time, you can get estimates of growth rates, long-term catch series over time, the abundance of fish through time, as evidenced by the numbers being tagged. And we can look at backcast environmental influences on those peaks and troughs in good years and bad years. And this has created a huge number of opportunities for research, we call mining the data. And here's just one example. This is a PhD student of mine, Tristan Guilliman, who I think is listening. G'day, Tristan. And this is a lovely graphic that he produced of the species composition of the 3 species of marlin, black marlin, black marlin in black, and you can see blue marlin in the middle and striped marlin in teal down the bottom. And you can see that these data series are the proportions of the 3 species caught in different latitudes over the entire time span, not just of the tagging program. And you can see that this goes back before the tagging program started. And what that data is records of fish that were landed and weighed by game fishing clubs right back to the 1930s in the case of the south coast of New South Wales. And so these clubs are very well organised, and they produce annual reports. So we're able to glean from the annual reports of many clubs the individual dates and weights of the 3 species of marlin going way back. And then we merged that into the tagging data as tagging took over from a capture. And here's another of Tristan's graphics showing, again, the same time series. And you can see the capture, the fish being captured going way back and the numbers. And then you see that in more recent years, a lot more fish being tagged. And just note the scale on the vertical axis there. You see that the tag and release numbers are orders of magnitude higher than the capture numbers, even though to see them we sort of plot them like that. So many more fish are being tagged in more recent years, but fantastic amount of data. And I mentioned environmental influences on abundance. So we've had a whole series of PhD and honours students come through. I think about 10 different universities now use this data. The data's already there. They don't have to go and collect it, but then they can be mentored by computer programmers and computer modelers to help them make models of how these environmental cues like sea surface temperature, chlorophyll, El Nino, La Nina, a whole range of environmental remote sensed information that's been available since the 1990s, and we overlay that with the tag release data, which is fantastic. And this is just a 2 pages out of, I think, about 8 pages of scientific reports and published papers and peer-reviewed papers that have used data from the tagging program over that period of time. So when the program started, it was not regarded as by some in some scientific circles as being very useful, but that's absolutely been proven wrong over over many years. The important thing here is that many anglers have been tagging fish for a long time, and they get used to being part of a scientific program. So that has spin-offs in cooperating in other areas as well. So these are just 3 areas: tournament catch per effort, how many fish are caught per boat day in tournaments, very important over a long period of time. Tournament sampling and research on landed fish, so making the fish that they catch in game fishing competitions available for research, and also non-lethal tissue sampling on released fish. So as I said, 95% of marlin are released, but we've been able to get tissue samples from those from anglers by taking fin clips. So just research and monitoring. This is a program I've been running now for about 23 years at New South Wales game fishing tournaments, again funded by recreational fishing licenses. And we make this available for researchers from different universities and even overseas. Scientists have visited and conducted a really large number of research projects on these fish because they're very difficult to get access to otherwise. And this is just some areas. Global genetics of many species. We've contributed to the global genetic studies of blue marlin, black marlin, striped marlin, yellowfin tuna, dolphin fish, even remoras, where the anglers have actually taken remoras off live fish and brought them in for global genetic studies, mako sharks, tiger sharks, etc. Movements, the pop-up satellite tagging is in the last 20 years has really been revolutionary, and I'll mention that again in a sec. But through tournaments, we've had a lot of satellite tagging done with no expense for using the boat. The anglers will be volunteering their boats to do that. Studying age and growth by taking hard parts, vertebrae from sharks and otoliths from billfish. Reproduction, of course, we sex every single fish that's landed at tournaments and look at their reproduction stage. A lot of physiology done on eyesight, olfaction, taking these fish coming quite fresh so that the physiologist can take really fresh samples to look at those sorts of senses of fish. Dietary studies, of course, what they've been eating, and look for muscle samples, stable isotopes to see what they've been eating over the last 3 or 4 months. And parasitologists love coming to tournaments and delving into the bowels of and the internal organs of these pelagic fish. And every time they do, they find new species of parasites, which is wonderful. Just a bit of a montage of the activity at this particular tournament every year in New South Wales at Port Stephens, setting up outdoor labs. I mean, there's a whole gaggle of scientists and students on the left setting up their lab, and the public are very interested in this. They come and watch, and we tell the public over loudspeakers what's going on and what sort of research is happening. Routinely measuring every fish, taking a flesh sample, and on the left there's 3 different scientific groups doing 3 different research projects on that fish. Sharks, tiger sharks and mako sharks are the 2 main species that come in at tournaments, and they have been utilised for lots and lots of different studies as well, which we won't go into in detail, but it's all in the literature. Parasitologists, as I said, they get a lot of parasites, and I think the lady in the middle is Shokafa Shamsri, who's now at Charles Sturt University in Wagga, and she's had, I think she came along very early in her career and started to look for parasites and since then she's become a leader in the field in world parasitology. And physiology, these students from Macquarie Uni and researchers are looking at the eyesight of billfish, very interesting results. And the non-lethal sampling. Sam Williams on the right did a PhD, I was his co-supervisor at Queensland Uni, And he needed samples of black marlin for global comparisons of tissue. And so the anglers at this particular tournament came in with these fin clips off the tip of the pectoral fin or off the tip of the dorsal fin. And we got about 130 samples split between Western Australia and Eastern Australia, all from anglers releasing the fish but bringing in the tissue sample. And Sam was able to show from that black marlin in the Indian Ocean and the Pacific Ocean were genetically distinct, had a different spawning area. Of course, we accumulate a lot of information just from routinely measuring, and the fish are all weighed on certified scales, and we sex all the fish. And so we've got this increasing database of just basic biological material information as well. But biological material, this is the tissue bank that's been accumulated. This is in the premises of Pepperell Research in a -80-degree freezer that was donated by a philanthropist, game fishing friend of mine who's been very active in research. And we've got well over 1,000 samples from all of those game fish species I mentioned before, but mostly billfish and sharks. And these have been used in many studies all over the world, and they're there for anybody to request the use of these samples. This is just one example of the scientific personnel at one tournament fairly recently. In that picture, there are 4 PhD students working on four different topics, some on sharks, some on billfish, from 4 different universities. So this has been a great training ground for postgraduate students, but also students just coming from high school into uni and getting hands-on experience in the field. And we do actually pay them through the funding we get from the saltwater licenses. We pay them an hourly rate to help as well, so they get some pocket money. And just 2 more examples of citizen science for spin-offs here. I mentioned pop-up satellite tagging. This has been fantastic. We've Dave Tomlinson, who's attending this seminar today, he was one of the first to make his boat available free of charge for us to go out and put acoustic tags on black marlin. First time that had been done. But since then, hundreds and hundreds of billfish have been tagged, and sharks and other species from recreational boats that have been made available free of charge, which is fantastic. And because the results of pop-up tags are fantastic, because those little red circles, this is black marlin on the left, by the way, they've been tagged mainly off Lizard Island, the Great Barrier Reef, and that's where they popped off over a number of years. The red little circles are where they pop up. And because the beauty of this is that not only does it tell you roughly an estimate of the actual path that the fish took, not just a straight arrow, but also they're fishery independent. You don't have to have somebody catch the fish to send the information back. So if the tag does pop up, it's independent of fisheries, so it can pop up anywhere in the world. And on the right-hand side, the pop-up tags also show the depths and temperature of the fish at 30-second intervals, and you can download that. So in this example, this is data from black marlin tagged off Eastern Australia. The bottom graph shows that the fish that are 30 kilos average estimate, the depth that they swim to over a daily period is much less than the bigger fish up the top, that they average about 150 kilograms estimate. And you can see that at night time, they rot up near the surface, and in the daytime, they're diving down to 300 or 400 meters. And this second little citizen science spin-off, daily aging of juvenile black marlin. We're very lucky in Australia to have juvenile baby black marlin come in shore, come in quite close to the coast, where they're accessible by mostly recreational anglers, because commercial fisherers can't take black marlin, they're not allowed to. And over the years, I've requested that some of these are kept for daily aging. And so the otoliths of fish, especially small fish that are quite very young, you can actually count daily rings rather than annual rings to find out how old they are in days. And so we have had these fish donated over, especially fairly recently, mainly from Hervey Bay, where in Queensland, where a lot of these fish are caught. And so they're sent to a lab in Victoria where they're dissected, and this is funded by the Game Fishing Association of Australia. And you can see the size of the otoliths. That's a 5-cent coin, for those of you who remember what a 5-cent coin looks like. And they're absolutely minute. And on the right is the, you can see the daily rings with this otolith that's been etched, and each one of those rings represents a day, we assume, and there's good reasons for assuming that. And so here's the results of these, just three of the fish down below. And so the smallest one that we've had donated so far was very tiny, only weighed 360 grams. That was estimated to be 66 days old, hatching from an egg about 1 millimeter in diameter. And so the larva on its first day would be about 2 millimeters long. So in 2 months, it's reached that size. 80 days, it's reached, well, some of that particular fish reached 70 centimeters, and the one on the right, 5 kilograms at about 4 months old. Something like that, 4.5 months. Very, very rapid growth rate. In fact, one of the fastest growth rates of any fish in the ocean. Communication education. This is something that Adam's very keen on. Reef Ecologic, his company does a fantastic job of communication education. I've always assumed ever since I left the fisheries department that this is vital to education and communication with recreational anglers, and I've been lucky enough to be able to do that through many different ways of doing it. This is the first, 2 months after I left the department, I contacted Modern Fishing magazine, and this is my first column called Taglines, and that continued for 25 years. I wrote a column every month for 25 years on something to do with fish biology, and I've also got busy on a lot of other magazines as well. So I ended up writing about over 850 columns, quite often answering questions from recreational anglers, and also 180 feature articles in fishing magazines mainly. And I think that got a lot of response. And it's very important with citizen science too to give feedback. And so these are the 2 annual reports that I write every year on those 2 programs. And that's put up online, and very detailed annual reports of what's gone on in those 2 programs. Lots of easy to read text, and layperson friendly graphics and that sort of thing. And just I thought I'd just throw up some books that I've put together over the years as well. And these are, again, very, very important in terms of communication and education. Now, I wanted to return, this is the sort of last part of the talk, return to this arrows on maps, I call it. Arrows on maps that you get. And over the years, when I looked at these arrows from black marlin that have been tagged in Australia and being recaptured in all these remote places, distant places, I started to think, well, who was at the other end of that arrow? Who sent that tag back? Who were they? And what's their story about marlin and billfish? Are they important to them? And why did they send the tag back? And what's their fishing culture? So in the early days, we used to get letters. Now we get emails, really, because the tag is printed with an address and an email address these days. But it used to be really exciting to get these letters and open them up, and they could be from anywhere around the world, and find some lovely letters about people who'd taken the time to send them back. And so I decided to go to the ends of some of these arrows and find some of these fishers and fishermen, and talk to them, and get their marlin stories along the way, and maybe write a book about it one day. And so I've been doing that for about 15 years, and these are the places that I've gone to so far, and found billfish stories. I've just got a couple of these examples. So this area off the tip of Papua New Guinea, the Milne Bay province, and some islands off there, we've had a lot of marlin recaptured from up there by village fishermen in dugout canoes, basically, and the tags have come back. So my first trip was up there in a game fishing boat, again, donated, the time and everything by a friend up there. Took us around to all these islands in that area. And the guy in the middle, turns out that marlin fishing is important to them, and but also competitive. So that the fishermen will each have a tree in some of these villages, and they'll impale the tail of their catch with the bill of the marlin to see who's the champion marlin fisherman, which is great. And the guy on the bottom right, Rodney, he came out to meet us and told us his marlin stories, and that's his game fishing boat at the back there that you can see in the water. And the guy on the left, when we were in Alatow, right on the eastern tip, we put out over the local radio that a request for anybody that had caught marlin to come in, and we mentioned some names. And this lovely man turned up, and he caught black marlin 15 years ago. Gow Holliker, his name is, and sent the tag in with his letter. And so 15 years later, he hears it on the radio, comes down and meets us. We gave him some fishing gear and got his stories, and he was pretty flabbergasted that I had a picture of his letter on my laptop computer. But this story was the ultimate, though, in PNG. I'd heard from a doctor, a doctor who worked in a hospital at Alatow. He was a humanitarian doctor. He told us that he'd saved the life of a man who'd been speared by one of these fish, a billfish. And so he said he knew the village where he came from. So we jumped in a four-wheel drive and drove about 50 miles through the jungle with an interpreter to this village. And we found this man who had been speared through the chest by a sailfish, as it turned out. He'd been fishing in a dugout canoe, had hooked a sailfish. The line had gone slack, and he was looking around, and because he's low to the water, and the sailfish had come out, jumped, and penetrated through the back on the left-hand side and come out his chest on the other side. And the doctor who treated him said the bill must have knocked his heart aside as it went through. He got an infection. He was in hospital for 2 weeks, and but he saved his life. 10 years later, I arrived, and he was a bit stunned to be talking to me, and told me his story. And I asked him if he ever went fishing again. He said no, never went fishing again after that incident. I don't blame him. But this is not an uncommon story of billfish being yes, very prestigious to catch, but also very dangerous, and they are feared in some areas because of that sort of thing. Second little place was Taiwan. Off this little star there is the town of Chiangong. Now it's billfish town. They have a billfish festival. It's a harpoon fishery actually that catches the billfish. They use other methods too, but again, a prestigious way of catching marlin and very skillful. And on the right there is a catch of the day from harpoon fishers. And the two guys on the bottom left are two of the harpoon fishers. And they had some great stories. They both, by the way, are missing the top joint of their left thumb in separate accidents. The rope on the harpoon when the marlin sort of escaped got wrapped around their thumb. And so they belong to a rather exclusive one joint thumb fishing club of Chiangong. They have a marlin festival there too, and a marlin temple. And it's quite an extraordinary place, and lots of stories. And lastly, just I mentioned the Western Australian recaptures from India and Sri Lanka. In India, I found the lady who had sent the tag back from Pondicherry in India. On the left, that's her. She was the deputy director of the fisheries department there, and it made the news in that part of the world that there was a tag from Australia. And again, about 10 years later, I tracked her down and was able to have a really good chat with her, and we were both delighted with the fact that this one fish had kind of brought us together. And I was shown around a lot of the fishing ports by the guy in the check shirt who was a billfish scientist. Through this, I'd found him through scientific papers, and he gave us guided tours and interpreted for us with the fishermen with their stories. And lastly, in the southern Sri Lanka, we found the fisherman who had returned the tag there. His name is Samira, and that's his fishing boat on the right. And he invited us to his house, and we had a lovely seafood meal, and we gave him lots of fishing gear as well, and we're very happy to hear his stories. And And the other guy there with the smiling face is my compatriot John Diplock, ex-fisheries scientist, who accompanies me on all these trips. He likes the adventures too. Just in case you had any doubts about my obsession with billfish, I just thought I'd put this little collage together of if I walk past anything to do with the billfish, I've got to have my photo taken in front of it. And so this is over the years. And that's it. I just wanted to thank all of the citizen scientists who've taken part in all of these studies, and may it continue for a long time. Thank you very much.
Well, thank you very much, Julian, for sharing that story. 50 plus years of passion with billfish. There's quite a few questions on the chat, and I know Mal Poole, you've probably got a few questions. So let's start with you. Julian, if you want to just, everyone can put their video back on and ask Julian questions. This is a fantastic opportunity.
Hey, Julian, how you going?
Hey, Malcolm.
Well, great talk. Thank you very much, and thanks to Adam for organising it. Impressed. I saw a question up there. How many billfish have you caught, Julian?
Oh, a lot. Probably about a lot of sailfish. Sailfish are pretty easy. I've caught my first black marlin was caught up off the Barrier Reef in the late 1970s when I went up to Lizard Island, funded by some philanthropists from New South Wales to go up and see if those fish were breeding. And I was lucky enough to go out on a big game boat that was donated to me for the day by the angler from America who had a hangover. He said, "Oh, you just take the boat out." So I went out with a famous fisherman, Peter Bristow, and caught my first 2 black marlin. Yeah, probably about, I don't know, about 100, but let them all go, of course, with tags in them. But of course, I've been on many, many game fishing boats all around the world and seen probably hundreds and hundreds of billfish caught and tagged and released mostly, and pop-up tagged as well.
No worries. Look, I threw a couple of questions up early in the process. Obviously, citizen science is a great thing to have. How has this data enhanced fisheries management around Australia and the world? Another question would be, what could we do better? Another question is, how do we better use such data in relationship to proposed offshore wind farms like at Port Stephens and elsewhere? And what about limiting the misuse of such data?
How long have you got? What was the first bit again?
How has this citizen science data enhanced fisheries management around Australia and the world?
Oh, yeah. Okay. So the data is used even in stock assessments, which is really important on looking at the sustainability of some of these species. The historic club data and the tag data is used as a surrogate of catch. And also the tournament catch effort data is used as well. Growth rate is used in stock assessments. Well, I mean, yeah, one of the areas was showing the importance of the economic importance of the fishery, if you like. And that's what led to the ban on capture of black and blue marlin by commercial fishers. So it became those two became recreational only species in Australia. I don't want to go on, I don't want to give a really long answer to all of this. How can we do it better? Yeah, I don't know. I'd have to really think about that. I think it's done pretty well. I mean, we are engaged. The recreational community fishery is engaged and fishery management of these species, by the way, is at a Commonwealth level, really, the federal level. The irony here, of course, is that recreational fishing in all of our states is managed by state governments who don't always take a big interest in offshore, the distant species. In Queensland, for example, there's not much interest from the government here, but I live in Queensland, but in New South Wales there is, so that most of the researchers come from there. So we could probably do better by just stressing the importance to other state governments. Offshore wind, controversial. Yeah, look, the release information has shown where billfish are caught, and the offshore wind footprint off Port Stephens, for example, which is a really hot marlin area and has been for nearly over 100 years. the data from the tagging program shows that those areas are very important to historically where marlin are caught. Whether that's going to make a difference to where they might put these offshore wind structures, I don't know. The big thing there, though, I think with the offshore wind turbines is how close would you be allowed to fish to them, because they will attract bait fish and they will attract billfish, and so that answer, I haven't heard a definitive answer on that. I hope that sort of covers some of that, Malcolm, but yeah.
Yeah. So Malcolm,
Julian,
that was your bag limit of questions. You had 5. Everybody else gets 1. So I'm going to throw it open to someone else.
Sorry, Adam.
Anyone else? Andy, Sue, anyone like to ask Julian a question? Great opportunity.
Can I go again?
Hold your horses, Mal. We'll give him a chance, mate.
All right. Okay. Sorry.
Hey, this is Stu. Can I ask a question?
Yeah.
Stu?
Hey, how many large female black marlin have been satellite tagged? So, is there any that we know where they go once they've bred? You know, they come into Queensland and then like have their breeding cycle, but where do they go after that, and do they return?
Yeah, really good question. So before pop-up tags, we had quite a lot of the big female fish tagged. They're in the minority up on the ground. So I mean, for every 100 marlin tagged, probably 10 would be big females. We never got one of those recaptured away from the grounds. We had them recaptured back on the grounds after 1, 2, 3, 5 years even. But with the pop-up tags, there's a program called the Great Marlin Race, which is run by Stanford University, Barbara Block team there, and IGFA, the International Game Fish Association. And over a period of about 8 years, at the Lizard Island Game Fishing Tournament, something like 100 black marlin were tagged with pop-up tags donated by 2 philanthropists there, 2 very rich fishers who donated the tags. And in a couple of years, a number of big fish were tagged. When I say big, over 400 kilograms estimate, so up towards 1,000 pounds or more. And those have popped off right across the Pacific, which is really interesting. After, like, in the same year, well, within say 6 months, a number of them popped off in the Central Pacific, and one came off about 1,000 kilometers short of Chile, in the Southern Hemisphere, still off the Chilean coast. So that was really exciting, because that all happened in 1 or 2 years. The other years that they did pop-offs, they tended to stay around the Coral Sea. And so it just might have been in those 2 particular years that we got the conditions were right for the marlin to go that way. We know that the black marlin spawn up off the Great Barrier Reef every year between really October, November. But we're pretty certain there's no spawning ground on the other side of the Pacific. Black marlin do occur there. The world record was caught off Peru in 1953, believe it or not. But there are also black marlin caught off Panama and Guatemala and Costa Rica, quite not that commonly, but some years better than others. But we think that if they go over there, it's probably for feeding more than, it's certainly not for spawning. And do all the marlin do that, or just some? My feeling is that these billfish tend to scatter. If it's like a starburst, if you tag a whole lot, they go off in all different directions. It's not like we're sort of familiar with, say, bird migration, where all of the birds all go to one place and then they all come back again. Fish are much more random, and so not every individual does the same thing every year. But that's why we're still putting tags in and still trying to work out the puzzle.
Thanks very much. We've had some opposition to tagging large females just recently, and where would you stand on that? Is it safe to tag them, or is it do you think no tagging?
Sorry, I was just reading something else. So just that first part of the question. Sorry.
Hold on.
Julian, the
Yeah, I
question was,
So,
we've had some opposition from fishers for tagging large female marlin. What's your view on that?
Okay, so that relates to post-release mortality, really, and it billfish. Well, again, the pop-up tags. I should have mentioned this. The pop-up tags are a really good tool to find out how many fish die after release, because if the tag pops up, it will pop off. If the fish dies, it's set to pop off if the fish doesn't change depth, for example. And so from the data you get from the tag, you can tell if the fish died or not. And so with the larger fish up off the barrier reef, the survival rate has been good. It's probably about 80%, I think, from memory, 80 to 85%. So I would be encouraged, and we think that shark depredation is one of the main factors in post-release mortality of black marlin up off the reef. And, you know, when we were acoustic tagging marlin there, we tagged 8, I think, and one of those was definitely chewed by a shark and swallowed the tag, and we followed the shark for 2 days. Until we really, yeah, you remember that, Dave? Yeah. No, but having said that, the other big change in this fishery has been transitioning from J hooks, which tend to hook the fish deep, to circle hooks, which mostly hook the fish in the corner of the mouth and have a very high survival rate, especially when there's no sharks. In fact, there was a study done in the United States on white marlin, which are admittedly quite small marlin, but they had 100% survival of 20 white marlin tagged with circle hooks, and the object of that exercise was to tag all of the fish that were caught, not just the ones that looked healthy. So we're pretty confident that marlin are quite robust. They survive well. Even we did tag a big marlin with an acoustic tag, and it was belly up, and we thought it was probably going to die, and it revived after a couple of hours, and then we tracked it for another day and a half, and it was doing all the normal things. I hope that answers the question.
Great. Question from someone else.
I've got a question.
Yeah, go, Mark.
Great talk, Julian. Thank you very much. You mentioned the babies are inshore, the larvae are about 2 millimeters long, rapid growth rate, and October to November spawning. That's great. I'm just wondering whether the actual spawning event has ever been observed.
Oh, good. Yeah. Anecdotally, yes. From quite a lot of the charter captains in that fishery, especially the long-term charter captains, many of them have seen the marlin, a big marlin on the surface, either following their bait or coasting down waves, or in the swell, followed by 5 or 6, that sort of number of smaller fish, which are almost certainly males. And yes, and they have seen cloudiness in the water with the males come really close to the female, and I'm pretty sure they have seen the event take place. Yeah. I don't know if it's more nocturnal or diurnal, and the observations that I've had of that are few and far between. But yeah, and even to the extent that that image is known quite well, so that some of the great billfish artists have painted that image, the 4 or 5 little black marlin very close to the female. Yeah.
Be awesome to get some footage of that.
Yeah, it would be. And I often get asked, I mean, if they're producing, we've done fecundity counts on this. When the first trip to Lizard Island, the fecundity counts were like 65 million eggs to 220 million eggs from a dozen fish over 1,000 pounds. And of course, people say, well, are all the eggs fertilized? Well, probably not, but evolution would suggest that the efficiency would be fairly good, for the males to get as close as they could, and when the event happens. And yeah, it's and of course we're pretty sure that marlin, like tuna, don't spawn all their eggs all at once. That's highly unlikely. That they're spawning every second day, every day, every second day. a portion of those eggs. So it might be millions of eggs every day for a period of a month or something like that, which the tunas certainly do that.
Interesting. Thank you.
Thanks.
Okay.
We might take 2 more questions from people who haven't asked a question yet.
Hi, Adam, and hi, Julian. Lovely to see you again after all these years. In terms of, I guess, a couple of questions about the citizen scientists themselves. So how many, approximately, I realize that it's like this program is over decades. So how many citizen scientists have been involved, number-wise?
So in terms of numbers of people tagging fish,
Yeah.
60,000, more than 60,000
Wow.
over that period of time. It's a long period of time, but in any given year, there'd be, well, we get, say, 10,000 fish tagged in a given year. And let's say, you know, a fisherman might tag, say, 5 fish. Some fishermen have tagged hundreds of fish, but what's that, like 200 anglers a year, or no more than that?
About 2,000.
2,000, that's right. And that'd be about right. I mean, there's members of game fishing clubs in Australia number up towards 10,000, and not just members of clubs tag, charter boat operators. Like each of the charter boat operators up off the Great Barrier Reef, when they tag fish, they might be tagging well, about 800 fish a year up there. So with all their clients, maybe hundreds of anglers would be tagging. Yeah, it mounts up. So yeah, that's a good question. And in terms of the other sort of activities, certainly hundreds, if not thousands, over the years.
No, fantastic work. I mean, obviously I live down in the Wide Bay, so for me, hearing about the young marlin down in Hervey Bay was fascinating, because I know we've got a lot of game fishers in our area. So to hear that not only are they spawning in GBR, they're coming down into the Bay Area to grow out, and the fact that they scatter it, just sort of this vision of very similar behaviour by marine turtles. So you know, yeah. As always.
Yeah, no, thanks. It's good to see you too. Yeah.
Yeah. Thanks, everybody.
Thanks. And we'll take one final question before we wrap up.
Yes, I've got a question for Julian, if I could, please. So, hi, Julian. It's been a few years. We met a long time ago. Just on citizen scientists and keeping them engaged. So, I've noticed and spoken to a fair few wreck fishermen on the south coast of New South Wales lately, and they're seeing a big pattern this year. A lot of people are moving away from contributing to tagging, and more so the wreck fishers are focused on catching numbers of fish, and they're fishing a little bit differently. And I'm based down here in Victoria. We've seen a similar sort of pattern in recent times on our southern bluefin tuna, where there's been a little bit of a drop-off on citizen science engagement. I'm just wondering if you had any ideas on how we can turn that around, both for billfish and other pelagic species.
Yeah, I think it's a good question. I think it's a bit cyclic. You know, the younger anglers coming through quite keen on tagging and getting the information. People who've done it for a long time and haven't had any fish recaptured, for example, they kind of go, well, I've been there, done that. Different. If it's club based, they get points for it. So there is a competitive kind of element. If people aren't in clubs, it's not that competitive, and you know, they might want to catch fish to eat and that sort of thing, or just release fish. There is a trend away from tagging in certain fisheries, like sailfish off Broome and black marlin up off the Barrier Reef. We are trying to get those fishers to fill in exactly the same sort of card that the tag comes with, but a release card, so that we at least get the information of that fish was caught at that place at that time, and we're encouraging that to happen. In terms of southern bluefin tuna, by the way, with the engagement of anglers, there is a fantastic program that is still going called Tuna Champions, and it was run by Sean Tracy, Professor Sean Tracy out of UTAS, and they really heavily engaged with clubs. They went around to talk to many clubs on how to improve the survival of some bluefin when you were releasing, regardless if you were tagging them or not. And also, if you're going to catch them, how to dispatch them humanely and treat them well so that you bleed them properly, so that the food that you're going to get from it is in tip-top condition in terms of icing and all that sort of thing. So that was a very successful program. But yeah, you're right. Continuing the engagement is the difficult part, and yeah, you need those programs to do that, and they come and go. Those programs, yeah.
Yeah, for sure. That Tuna Champions program you mentioned is fantastic. I'm actually one of the ambassadors for it down here, and we've seen it. It's basically been an exponential increase since it come in, and we just need to keep that going now that that program's potentially slowing down. So,
yeah, I agree. Yeah.
Cheers.
So it's 11 o'clock. Thank you very much, Dr. Julian Pepperell, Dr. Fish. That was a fabulous presentation, not only on your passion for pelagics, but also your interest in the people, the citizen scientists throughout the world who have contributed to the knowledge of these amazing, beautiful species. Thanks everyone for your questions. We will share a recording of this presentation. And in order to continue storytelling, we are planning a monthly webinar. The next one's being delivered by Bill Sornock from InfoFish. And then we've got one on shark depredation in the future by Dr. John Mitchell. If you've got any suggestions, we are trying to encourage a range of people, from traditional owners, commercial fishers, citizen scientists, rec fishers, to tell their stories. So contact us. This is really important, and we thank everyone for being online today.
Thanks very much.